Welcome friends to a new session on genetics. In this session, we will discuss extensions to Mendelian inheritance, gene interactions and other modified dihybrid ratios. Let us see how gene interactions can influence a particular characteristic. According to Mendel's experiments, a pair of genes controls each character, but later discoveries proved that the interaction of more than one pair of genes controls a single character. This is known as interaction of genes or factor hypothesis. The interaction of genes was proposed by William Bateson and Reginald Crundell-Punnett. The key diagnostic is that these interactions produce modified dihybrid ratios. There are various types of interactions and these interactions leads to a different range of phenotypes. Inhibitory factor is another important intergenic or non-allelic gene interaction, but in Pliotropism a single gene contributes to multiple phenotypic traits. Now let us understand about inhibitory factor. As we know, the interactions of genes may involve two or more pairs of genes. In symbol, we could say that expression of one gene depends on the expression of another gene in gene interactions. These interactions produce modified dihybrid ratios. Inhibitory factors involves two pairs of non-allelic genes and it affects the same character. In this, one of the dominant genes suppresses or inhibits the expression of other dominant gene and this inhibiting gene has no visible effect at all. Example, leaf color in paddy. The occurrence and distribution of anthocyanin pigmentation in the leaves of the rice plant is very variable and a very striking feature of the crop. The genetics of this character has attracted wide interest and attention from early time onwards and its mode of inheritance in the individual plant part has been extensively studied. Many geneticists reported the number, role and interrelationship of the genes responsible for the expression of anthocyanin pigmentation in the leaf, which may entirely non-pigmented or fully pigmented. In the later case, the leaf blade may be purple, purple wash or faint purple wash in color. Purple leaf blade has been shown by the interaction of a basic and two complementary genes and all the three genes are essential for the production of purple color. The phenomena is known as autumn leaf color. It is a phenomenon that normally affects the green leaves of many deciduous trees and shrubs take on various shades of red, purple, yellow, black, pink, orange, blue, magenta and brown during a few weeks in the autumn season. The phenomenon is commonly called autumn colors or autumn foliage in British English and fall colors, fall foliage or simply foliage in American English. The purples, reds and their blended combinations that enrich autumn leaf color come from anthocyanins in the cells. These pigments are actively produced at the end of summer and not present in the leaves throughout the growing season like carotenoids. The presence of anthocyanin color in rice is determined by the complementary action of two genes capital C and capital A. The gene C is the basic gene for chromogen production and gene A exerts its activation effect on gene C and convert the chromogen into anthocyanin. The gene C and A comprises multiple allelic series of genes. 3 alleles have been found at A locus and 5 at C locus. They are arranged in the order of dominance as shown in the screen. The expression of anthocyanin color is attributed to the complementary effect of the genes C and A. For distinct coloration in paddy, the presence of C and A and another gene P exists. This is concerned with the distribution of chromogenic substance. The majority of varieties, as a matter of fact, however, possess the gene P in common. As a result, a combination of any of the alleles of C and A loci, it follows the principal color types on the leaf. The expression and occurrence of color in the leaves are closely related to the constitution of genes. The occurrence of coloration in these parts when the genes coexist with any gene combination at A and C loci. When exposed to direct sunlight, the PI gene distribute the color over the entire surface of leaf sheath, leaf blade, collar, ligule, auricle, internode, node and pericarp also. The PN gene distributes the color in leaf margin, leaf apex and entire surface of node, auricle, collar and ligule. The most striking colored parts with the PN and PI are the node and leaf blade respectively. The expression of PI gene is reduced by the presence of a suppressor gene JPI that inhibits the distribution of colors at the center of leaf blade. Thus, at present it may be more natural to consider that the appearance of the new type may depend on the presence of another alleles at the C and A loci. There are multiple alleles at 2 loci, 6 alleles at the C loci and 4 at the A. They are temporally designated as CBN and AE respectively and may be arranged as shown in the screen. As to the action of CBN and AE, CBN may be less potent than CBR and give no sign of anthocyanin coloration when coexisting with A. However, when CBN coexists with AE, the highest potent allele at the A locus, also AE is estimated to have more activation effect than A faint red color is produced. Now, let us look into Pliotropism. It is a phenomenon in which a single gene controls a number of phenotypic traits in the same organism that can arise in a variety of ways. Otherwise, it is a condition in which a single gene may have 2 or more phenotypic expressions and it affects evolutionary rates of alleles and gene frequencies. These pleiotropic effects or phenotypic expressions often seem to be unrelated to each other. Pliotropism is a central term in developmental genetics. The term pleiotropic comes from the Greek word plio means many and tropine means influencing. Pleiotropic genes are the genes which, in addition to their main effect, may also act as modifiers for another but entirely different gene. Such genes having more than one effect are called pleiotropic genes. 100 years ago, the term pleiotropism was formally introduced into the literature in 1910 by the German genetist Ludwig Hermann Plate, who was a disciple of Ernst Haeckel. He wrote a thorough and extensive defense of Darwinism, but before Mendel's work had been assimilated, pleiotropy had an important influence on the fields of medical genetics and physiological as well as on evolutionary biology. Mendel described an early case of pleiotropy in his classic 1866 paper. His character number 3 for Propism displays either violet flowers, brown seed coat and axial spots or white flowers, white seed coat and lack of spots. Mendel states that these three characters assigned to each strain are always found together, and he considers them to be correlated and under the control of a gene. Mendel believed that the three characters, that is flower color, axial spots and seed coat color, are due to a single gene, and he considered this sort of inheritance, though in a rather superficial manner. Now, coming to this figure, it shows the multiple phenotypic effect of a single gene. Here you can very well see about Pliotropism. A single gene is influencing multiple traits, trait A, trait B as well as trait C. Now, let us look into some examples of Pliotropism. 1. The AY allele in mice has two phenotypic effects. It controls lethality and yellow coat color. A second one is Marfan syndrome. In short, it is called MFS, an inherited connective tissue disorder in human beings. It is characterized by slender body, long limbs, lens dislocation, susceptibility to cardiac diseases and hypermobility of joints. All these are caused by a single pleiotropic gene. The pleiotropic manifestations involving primarily the ocular, skeletal and cardiovascular systems, some of which are life threatening. Marfan syndrome has historically always been considered as a condition caused by the deficiency of a structural extracellular matrix protein fibrillin 1. The study of Marfan mouse models and Marfan related conditions has shifted our current understanding to a pathogenic model that involves dysregulation of the cytokine transforming growth factor beta. In short, we call it as TGF beta signaling. A third example is sickle cell disease. In short, it is called SCD, also known as sickle cell anemia, and drepanocytosis is another example. It is a hereditary blood disorder, genetically transmitted hemolytic disease. In this case, homozygous have two copies of abnormal genes that code for beta hemoglobin, leading to the formation of sickle shaped or crescent shaped hemoglobin with very low oxygen carrying capacity. As a result, it impairs circulation. This often leads to thrombosis, cardiac failure and renal failure. Thus, in a gene, a single mutation has multiple consequences in numerous tissues. The pleiotropic effect is associated with a number of acute and chronic health problems such as severe infections, attacks of severe pain and stroke, and there is also an increased risk of death. A fourth example is one of the most important example in pleiotropy in human beings is phenylketonuria, an inborn metabolic disorder. In this, the homozygous recessive individual lacks the enzyme phenylalanine hydroxylase needed to change phenylalanine to tyrosine. The abnormal autosomal recessive gene on chromosome 12 causes the lack of enzyme phenylalanine hydroxylase. The disease symptoms in children are decreased pigmentation of hair and skin, eczema and mental retardation. The heterozygous individuals are normal, but they are carriers. Towards the left of the figure, you can see various symptoms associated with phenylketonuria. The upper part shows a case in normal person wherein the phenylalanine is converted into tyrosine, which further converts into tissue proteins, melanin, etc. And in the lower portion, phenylalanine is not converted into tyrosine, instead it gets converted into phenylpyruvate and further gets converted into phenyl acetate as well as phenyl lactate, and this will lead to a phenylketonuria condition in the patients. There are 7 types of pleiotropy. The pleiotropy can be basically classified into 7 types, which is given in the table. These have different levels of importance and significance. All these types of pleiotropy carry a long term retribution in terms of evolutionary constraints. A gene controls more than one biological function is more limited in its envelope of possible variations than is a monofunctional gene. Moreover, alleles that is selectively advantageous for function in one tissue may be disadvantageous to function in another tissue. Here you can see a table wherein classification of different types of pleiotropy is given. The first column gives the type of pleiotropy, the second column gives the situation, and in the third column the example is provided. Now let us look into each type one by one. The first one is artifactual pleiotropy. In this case, a single mutation can affect more than one process because 2 genes are located next to each other in the genome. An example is the claret non-disjunction alleles in Drosophila, which affect both meiosis and eye color. The claret locus of Drosophila melanogaster comprises 2 separately mutable domains, one responsible for eye color and one responsible for proper disjunction of chromosomes in meiosis and early cleavage divisions. There are 3 types of alleles. 1. Alleles of the claret type that affect eye color only. 2. Alleles of the claret non-disjunctional type that affect eye color and chromosome behavior. And 3. A meiotic mutation non-claret disjunctional that affects chromosome behavior only. The behavior of claret alleles was cleared when it was cloned. There is an adjacent gene which encodes a kinesin molecule required for normal chromosome disjunction at meiosis, and the pleiotropic alleles are small deletions that affect both transcription units. Therefore, the multiple effects of such mutations are artifacts of the process of mutagenesis, hence the name. Especially the organisms with compact gene dense genomes are susceptible to artifactual pleiotropies. Now let us look into the second one, that is secondary pleiotropy. This is equivalent to relational pleiotropy. It describes the situations where a simple biochemical abnormality has multiple phenotypic consequences, sometimes with connection to the initiating mutation. Long-lived complex organisms, especially those with advanced abilities to compensate for physiological dysfunction, exhibit secondary pleiotropies. For example, one of the first human mutants is provided by the disease phenylketonuria to be understood at a biochemical level. In this case, a defect in phenylalanine hydroxylase, the liver enzyme, that leads to excess plasma levels of phenylalanine. In the brain, it affects myelination of axons and finally to Mendel retardation. Similar or more complex chains of developmental and physiological consequence can be seen in many genetic disorders in humans, such as the complex syndromes arising from thalassemias. Rudimentary mutations affecting pyrimidine biosynthesis, a familiar example is provided in Drosophila. Some of these mutations lead to defective development of the wing, but not of the rest of the fly, because pyrimidine levels become rate limiting for cell division in the rapidly proliferating tissue of wing disc, but not elsewhere. The third one is adaptive pleiotropy. It is otherwise known as exaptational pleiotropy. In this, a pre-existing protein has been clearly exapted or co-opted in evolutionary terminology to implement an additional function that is not related to its original biochemical role. An example of this type of pleiotropy is crystalline proteins, which is the most abundant proteins in lens tissue and play a refractive and structural role. It is a protein that in humans is encoded by the CRYGD gene. 4 gamma crystalline genes, gamma A through gamma D, and 3 pseudo genes, gamma E, gamma F and gamma G, are tandemly organized in a genomic segment as a gene cluster. In many processes, this protein acts as familiar metabolic enzymes such as anolase and LDH. Now, let us look into parsimonious pleiotropy. Parsimonious pleiotropy encompasses cases where the same enzyme catalyzes the same chemical reaction in multiple different pathways. Example, human guanylate binding protein, in short we call GBP1. GBP1 is a multi-step regulator of CAMP-PKA signaling, inhibits PKA downstream of GPA2P and CYR1P, thereby increasing CAMP dependency, promotes ubiquitin dependent proteolysis of IRA2P regulated by G alpha protein GPA2P. GPB1 has a paralog GPB2 that arose from the whole genome duplication. Complex metabolic consequences occur because of the loss of this enzyme. For example, at several steps in the biosynthesis of isoleucine and of valine, the same proteins are used. So, in bacteria, knocking out any of these enzymes leads to simultaneous prototrophy for both amino acids. In such cases, there has probably been no pressure to evolve independent regulation of the two pathways, either at the enzyme level or at the gene level. So, the organism utilizes the same protein in each pathway. Now, let us look into fifth one, opportunistic pleiotrophy. It is related to the two preceding types. A regulatory protein is recruited to perform an additional function in a different tissue. Example is the cis-B ASC genes acting as numerator elements on the X chromosome of Drosophila. In Drosophila, sex is determined by the ratio of X chromosome dosage to autosomal chromosomal dosage, and a few sites on the X chromosome act early in development as major numerators in setting this ratio. The sisterless A, in short called as cis-A gene, functions as one of 4 X:A numerator elements that set the alternative male or female regulatory states of the switch gene sex lethal. In somatic cells, cis-A functions specifically in sex determination, but its expression pattern also hints at a role in the yolk cell, a syncytial structure believed to provide energy and nutrients to the developing embryo. By genetic criteria, cis A and cis B are the most potent zygotic determinants of X chromosome dose. Now, let us look into the sixth one, combinatorial pleiotropy. Combinatorial pleiotropy applies in many of cases where a single protein interacts with a variety of different components in different cell types, and it altered biochemical activity and specificity in each different situation. This protein will have potentially diverse and multiple effects on a variety of tissues when affected with mutation. The same combinatorial strategy is observed even in unicellular eukaryotes. For example, in Cyanorhabditis elegans gene UNC86 is required in several distinct neuroblast lineages for daughter cells to become different from their mothers, and is also required for the specification of particular neural identities. Another example, the proteins that control mating type of yeast include the DNA binding protein encoded by MAT alpha 2, which has different functions in diploids and alpha cells. In diploid cells, it acts in combination with MAT alpha 1 to repress an additional set of haploid specific genes, but in haploid alpha cells, it acts to repress alpha specific genes. Now, let us look into the case of seventh one, unifying pleiotropy. Unifying pleiotropy is the final type because in ultimate biological output, it describes cases where the multiple functions of genes or locus are all related, but the immediate chemical functions are diverse. The different enzyme activities, structural components or binding domains are included in the same polypeptide chain or encoded by adjacent cistrons under common regulatory control. Otherwise, mutation of the locus can have complex physiological consequences. For example, in Cyanorhabditis elegans, the gene CHA1 is the structural gene for choline acetyl transferase, the enzyme which synthesizes acetylcholine. CHA1 is a complex gene which includes in UNC17 locus. It has been hypothesized that a single protein is encoded which consists of several discrete structural domains. Now, let us summarize the foregoing discussion of gene interactions. Genetic analysis can determine that the gene interactions determine the phenotype of an organism. Genes are located on chromosomes, come in pairs, so genes also exist in pairs. A gene can express itself in the homozygous state or heterozygous state as dominant or recessive conditions. According to Mendel, each character is controlled by a pair of genes, but later discoveries proved that a single character is controlled by more than one pair of genes known as interaction of genes or factor hypothesis. Gene interaction is of two types, allelic gene interaction and non-allelic gene interaction. Allelic gene interaction occurs between the two alleles of the same gene, but non-allelic gene interaction occurs between the alleles of different genes. Inhibitory factor is another important intergenic or non-allelic gene interaction, but in Pliotropism, a single gene contributes to multiple phenotypic traits. Inhibitory factor involves two pairs of non-allelic genes and it affects the same character. The occurrence and distribution of anthocyanin pigmentation in the leaves of the rice plant is very variable and a very striking feature of the crop, and it is an excellent example of inhibitory factors. Purple leaf blade has been shown by the interaction of a basic and two complementary genes, and all the three genes are essential for the production of purple color. The phenomenon is known as autumn leaf color. The presence of anthocyanin color in rice determined by the complementary action of two genes C and A. The gene C is the basic gene for the production of chromogen, and gene A exerts its activation effect on gene C and convert the chromogen into anthocyanin. The gene C and A comprises multiple allelic series of genes. 3 alleles have been found at A locus and 5 at C locus. They are arranged in the order of dominance as Pliotropism is a phenomenon in which a single gene controls a number of phenotypic traits in the same organism and was formally introduced by the German genetist Ludwig Hermann Plate. There are 7 types of pleiotropy: artifactual, secondary, adoptive, parsimonious, opportunistic, combinatorial and unifying. Phenylketonuria is one of the most important examples in pleiotropy in human beings. Now, let me give you some assignments from the portions we have already discussed. The first assignment is explain the biochemical basis of phenylketonuria and sickle cell disease. The second one is write short notes on different types of pleiotropy. Third, what is pleiotropic gene? How these genes modifies or control a number of phenotypic traits. The fourth assignment is explain the expression of anthocyanin pigment in paddy and the complementary action of genes responsible for it. And the last assignment is briefly explain how Mendel described an early case of pleiotropy in his classic 1866 paper. Now, I will give you some references for your further reading. The book with the title iGenetics, a molecular approach plus mastering genetics with e-text, 3rd edition, authored by Peter J. Russell, published by Pearson in 2011. Second, a book with the title An Introduction to Genetic Analysis, 7th edition, authored by Griffiths, Miller, Suzuki, Levontin and Gilbert. It is published by Freeman, New York in 2000. A third book is with the title Principles of Genetics, 8th edition, authored by Gardner, Simmons and Nestad in the year 2006 and published by John Wiley, New York. A fourth book is with the title Concepts of Genetics, 10th edition, authored by Klug and Cummings in the year 2011 and published by Benjamin and Cummings. A fifth book is with the title Principles of Genetics, authored by Gardner and Nestad in the year 1984, published by John Wiley, New York. A sixth reference is Developmental Genetics and Lethal Factors, authored by Hedon in the year 1961, published by John Wiley and Sons, New York. There are some websites also for your further study. First one is www.genetics.org. A second one is theagricos.com. Thank you for watching this session. See you next time. Until then, bye.